Uh, Good day, class. I hope you're fantastic and amazing. I'm amazing myself. So today we'll be focusing on DNA and meiosis, right? But firstly, we'll start with DNA, right? And the important points that you should know and remember is the history of the discovery of the DNA molecule, right? You should understand the parts or the roles that Watson, Crick, Franklin, and Wilkins played in discovering DNA, right? Who ended up getting the Nobel Prize, right? Who discovered the X-ray structure of the DNA, right? So it's important for you to understand those things. The second point is that you should understand the structural differences between the oxyribonucleic acid and ribonucleic acid, right? So you need to understand the structural differences between the two, right? So we're going to get to that in detail today. And the third point is that you need to understand the differences between DNA, mRNA, and tRNA, right? So those are the differences that you need to understand. The fourth point is that you need to be able to differentiate between DNA replication and protein synthesis, right? Because a lot of people tend to confuse transcription and DNA replication. So it's important for you to understand the differences between the two. So our fifth point is that you need to uh, identify or learn the processes by heart, right? For example, you need to learn transcription and translation by heart. So it's important for you to know that. We're going to go through that, right? The sixth point is that you need to understand the importance of DNA profiling, right? And how do you apply DNA profiling, especially when it comes to paternity testing and identification of criminals, right? So you need to understand the application of that. And our next point is that our second last point is paternity testing involving both parents and the child, as I've alluded to it, right? Okay, and the uses of DNA profiling, right? Without further ado, let's start right away. So let's get to the structural differences between the DNA molecule and the RNA molecule, right? DNA in full is deoxyribonucleic acid, and RNA in full is ribonucleic acid, right? So you can already see observable differences between DNA and RNA. So you can see that DNA is double-stranded and RNA is single-stranded, right? Does that make sense? Does that make sense? And another structural difference is that the monomers, one of the monomers are different, right? For example, DNA, when it comes to DNA, we have deoxyribose sugar, but when you go to RNA, you have ribose sugar. Does that make sense? Does that make sense? I hope it does make sense, right? Now let's look at the length. As you can see, the length of both molecules. When it comes to DNA, the length of DNA is long, and when it comes to RNA, the length of RNA is short. I hope that made sense, right? And another structural difference between DNA and RNA is that DNA contains thymine, whereas RNA contains uracil, right? I'll repeat that again. DNA contains thymine and RNA contains uracil, right? And most of the time, you should understand this. When it comes to RNA, we're not speaking about thymine, we're speaking about uracil, right? Remember, we have four bases in total when it comes to DNA. We have thymine, cytosine, guanine, and adenine, right? But when it comes to RNA, we have cytosine, guanine, adenine, and uracil, right? We don't have thymine when it comes to RNA. I hope that is clear. Hope that is clear, right? Okay, let's continue. This is just a revision, right? And it will help you to be able to apply um, the code of life as a topic when it comes to question, right? Okay, so let's continue with the RNA structure. Let's look at the RNA structure. So the RNA structure is single-stranded. As you can see, it's single-stranded. And if it's single-stranded, definitely there's no need for hydrogen bonds. So for the fact that it is single-stranded, it means that there's no need for hydrogen bonds, right? Because it is single-stranded, right? And still, the monomers or the building blocks of RNA are called what? Nucleotides. They're called nucleotides, right? And each nucleotide is made up of ribose sugar, pentose sugar because it's formed by five, it's a five carbon sugar, hence pentose. The word pentose, pent means five, right? Because it's a five carbon sugar. Okay, so ribose sugar, right? Ribose sugar, phosphate group, and four nitrogenous bases. As we have alluded to it, right? So the nitrogenous bases are namely adenine, uracil, guanine, and cytosine, right? And you understand, by now I'm sure you should understand the pairing of bases, right? Adenine pairs with uracil and guanine pairs with cytosine, right? Okay, now let's look at three types of RNAs. So the first RNA we have is messenger RNA, or you can say mRNA. And it's important, guys, to know that whenever you're writing mRNA, you should use a lowercase m, not an uppercase m, right? It's important for you to understand that. So the first type of RNA is messenger RNA. Does that make sense? Messenger RNA. So what's the function of messenger RNA? The function of messenger RNA is to transport the message from the nucleus to the ribosome for purposes of protein synthesis, right? So it's just carrying the message. What message? The message to form proteins. It has a message to form what? Proteins. There's a message for a certain protein that will be formed eventually at the end of protein synthesis. I hope that makes sense. Okay, let's go to the second type of RNA. So the second type of RNA is transfer RNA, right? Transfer RNA or tRNA. So what's the purpose and what's the function of tRNA? The function of tRNA is that it brings the required amino acid and the coding or and the matching anticodons, right, to the ribosome for purposes of forming a protein eventually. Let me repeat that again, right? What's the function of transfer RNA? The function of transfer RNA is that it brings the required amino acid and the matching anticodon ribosome, to the ribosome. So that's the function of transfer RNA, right? Okay, number three, the third one is ribosomal RNA. So where do you find ribosomal RNA? So you find ribosomal RNA where in your ribosome, obviously, and also it's formed by which structure? This is important, right? You won't find it in your books, but I'm sure when you use other books, you'll find it. So most of the time, they'll tell you that ribosomal RNA is formed by the nucleolus, right? It's formed by the nucleolus, right? It's formed by the nucleolus, and its function is not examinable in grade 12, right? What you have to understand is the messenger RNA, transfer RNA, and understand their function, right? And where are they found? Where do we find the messenger RNA? So we find the messenger RNA within the nucleus, right? Within the nucleus, and it moves out of the nucleus where it gets into the cytoplasm, then attaches to the ribosome. Does that make sense? Okay. Then when it comes to transfer RNA, where do you find the transfer RNA? You find it in the cytoplasm. Find it in the cytoplasm, right? Okay, we'll go to protein synthesis and go over that. Right. Okay. Now let's go to the second process. To the first process, not second process, to the first process, right? In DNA. So the first process is DNA replication. Where does the stage take place? Right? When does it take place? Right? So it takes place in the nucleus and during interphase. We'll learn more about interphase when we get to meiosis. Okay. So the first point is that the first point in this process is that the DNA double helix. It's important for you to say DNA double helix. DNA double helix unwinds, so that's the first point. It's coiled, hence it is important for it to unwind because it's a coiled structure, right? And by the way, we call the natural shape of DNA double helix. It's a helical shape. Okay, let's go to the second point. So the second point is that weak hydrogen bonds between nitrogenous bases break, right? And two DNA strands, they unzip. Obviously, before the DNA strand can unzip, the weak hydrogen bonds, they need to break because they're weak, right? So they need to break for it to unzip. Unzip meaning separate. Okay, let's go to the third point. So our third point is that each original strand serves as a template on which its complement is built, right? So each original template, each original DNA serves as a template. It serves as a blueprint, right? It serves as a blueprint in order to form a complementary strand. Does that make sense? A complementary strand using free DNA nucleotides in the nucleoplasm. Using free DNA, remember, there are DNA nucleotides roaming around the nucleoplasm. What is the nucleoplasm? The nucleoplasm is the fluid that's surrounding or that's within the nucleus. That's within the nucleus. I hope that makes sense. Okay, let's go to our fifth point. So our fifth point is that this results into two identical DNA molecules. Two identical DNA molecules. Does that make sense? And each molecule consists of one original strand and one new strand. So another point that I will add is that the whole process is controlled by enzymes. The whole process of DNA replication is controlled by enzymes. Okay, let's go to protein synthesis. Protein synthesis. What is protein synthesis? Protein synthesis is the formation of proteins. To synthesize is to manufacture. So what are we manufacturing? We're manufacturing proteins. Does that make sense? And we have two processes that are involved in protein synthesis. The first process is transcription. And where does transcription take place? It takes place where? In the nucleus. It takes place in the nucleus. Does that make sense? Right? It does not take place anywhere else but the nucleus. It takes place in the nucleus. Okay. So let's go to the first process of protein synthesis. So it's similar to DNA replication, but don't confuse it. You'll see the difference, right? The first point in transcription says that double helix unwinds, DNA double helix unwinds, right? And weak hydrogen bonds between nitrogenous bases break, and two DNA strands unzip or separate. Right, then now the third point, this is the important point. You shouldn't confuse transcription and DNA replication. In transcription, it says one DNA strand acts as a template for the construction of mRNA in the nucleus. Whereas DNA replication says each DNA strand acts as a template. So there's a difference when it comes to the two, right? I hope that makes sense, right? Free attaching to free RNA nucleotides from the nucleoplasm. Does that make sense? This is an important point, right? It says free RNA nucleotides, whereas in DNA replication it says free DNA nucleotides. I hope you can remember that and understand that fact. Okay, let's continue. Free RNA nucleotides form the nucleoplasm from form mRNA complements the DNA in the. Why is this not complete now? Oh, okay, it's fine. Let me just start over again. Let's go to the fourth point, right? So one DNA strand, or the third. Let me start to the from the third point. Let me start from the third point so that I don't confuse you, right? So one DNA strand acts as a template to construct a new mRNA strand, right, in the nucleus, right, attaching to free mRNA or RNA nucleotides in the nucleoplasm. Does that make sense? Then after that, we have the movement of mRNA. So the movement of mRNA after the mRNA is formed, after it's done, after the process is completed, it moves out of the nucleus, right, into the cytoplasm, right, where it attaches to the ribosome. And you see this arrow. It's a representation, this red arrow is a representation of the movement of the mRNA, right? But you don't have to include it, right? You can look up a lot of memos, right? They don't include the movement of the mRNA. Okay, now let's go to translation. What happens after mRNA has attached to the ribosome? This leads us to the next stage, which is translation, right? So translation takes place in the ribosome, right? It takes place in the ribosome. Okay, so the site of protein synthesis, or the site of translation rather, is ribosome. So that's the site of translation. Site means the location where that process takes place. Okay, so let's start with translation. Translation. So translation now, it's where the message that was carried by the mRNA is being translated. To translate is to change one thing to another, right? You change from one form to another. So the message that was transported by the mRNA, now we are translating it. We are changing it to proteins, right? Okay, now let's look at the what happens in the stage called translation, right? Coded mRNA leaves the nucleus through the nucleopore. So this is the movement. So they've included the movement, right? Through the nucleopore to the ribosome. Then tRNA pick up amino acids and complement mRNA in the ribosome, right? And number 8, amino acids bond by a peptide bond to form a protein. I hope that's clear enough, right? And also, one thing that you should know is that 3 consecutive bases in the mRNA are called codons, right? I hope that makes sense, right? And 3 consecutive bases that are in the tRNA are called anticodons. I hope that makes a lot of sense. Okay, so I think we're done with this process, right? We're done with this process. Can you see now the difference between the structural difference between tRNA and mRNA, right? So tRNA is like it's shaped. It has a different shape, right, as compared to the mRNA. And how do you identify tRNA? You identify it by the amino acid at the top, at its head, right? It's carrying an amino acid at its head. Does that make sense? Okay. Whereas the mRNA is just horizontal. It's just a horizontal structure, right, carrying nitrogenous bases. Does that make sense? And this structure here, where it is attached to, is called what? The ribosome, right? So this is the site of what? Translation. Okay. Now let's continue. Let's go to DNA profiling, right? So what is DNA profiling? So DNA profiling is used as a technique to identify. It's a technique to identify the sequence of bases, right? So it's a technique used to identify sequence of bases. Does that make sense? Okay. So the nucleotides are separated from each other in order that they are found in strand of DNA. Right. The next point states that nucleotides appear as dark bands. Right. You can say bands or bars. So nucleotides, they appear as dark bars or bands. Does that make sense? Right. Then the DNA profiling is different from fingerprints. So it's different. So never confuse DNA profiling and DNA fingerprinting. So never use the word DNA fingerprinting because it's different. They're different. They're two different things, right? Okay, let's continue. Now I want us to focus on this question, right? So they're saying that the diagram below shows the DNA profiles of a girl, her mother, and 4 males, right? Okay, which is the girl's biological father? Already they gave us answers, yeah? Already they gave us answers. So it will be kind of difficult to answer this question without seeing the answer, right? Okay, so here the answer they're saying is C. I'll just tell you how they found the answer. Okay. So now let's look at the girl's DNA. So this is the girl's DNA. So the first thing that you do, you remove the mother's bars from the girl's DNA, right? So let's look at the matching bars, right? So look at the first bar in the girl's DNA matches with that of the mother, right? Can you see the first bar is matching? So we're removing the first bar, right? Now let's go to the second bar. So the second bar does not match with any of the bars that are in the mother's DNA. So let's go to the third bar. Let's look at the third bar. Also, the third bar does not match with any of the bars that are in the mother's DNA. So we leave the third bar. Let's go to the fourth and fifth bars. So we see that the fourth and fifth bars are matching with the bars that are in the mother's DNA. So we're removing those bars. We're removing the fourth and fifth bar. So we only left with the second bar, right? And the third bar. So now let's check. Now let's compare now, right? Let's look at the second bar and see which bars, right, match with the second bar with regards to the four males, right? Okay. So look at this first bar. So this first bar is matching with male one, right? And male three, right? Okay. But it does not match with male four and male two. So it does not match with male four and male two. Okay, so male one and male three are possible fathers at this point. We haven't made a conclusive decision. Okay, now let's go to the third bar in the girl's DNA. So the third bar matches with both male 2 and male 3, right? Matches both with male 2 and male 3. So now they said the answer here is C, right? They said it's male 3, which makes sense because both of the bars, right, two bars, right, that are left after eliminating the mother's bars, they match with that of male 3. So it makes sense. They match with that of male 3, right? So it makes a lot of sense, right? So male 3 is the father. So the first thing that you need to understand is that you're removing the mother's bars first. So you need to remove the mother's bars first, and the bars that you're left with, those are the bars that you use to compare, right, with the bars of the males, of the possible four possible fathers, right? Okay, let's continue. Okay, so we're done with DNA. So that's DNA profiling, right? So they left a lot of information in these notes, right? A lot of information, but I'll just add those information, that information rather. I'll just add that information. So information that is missing are the uses of DNA profiling. So the uses of DNA profiling is that it's used to identify dead bodies, it's used to identify relatives, used to identify criminals, right? And also it's used in paternity testing. Right? There are a lot of uses for DNA profiling, right? But you just have to know 3 to 4 uses, right? You don't have to know a lot of most of them. You don't have to know most of them, most of the uses. Okay. Now let's go to meiosis. Let's go to meiosis, right? So what is meiosis? Before we get into meiosis, what is meiosis? So meiosis is a cell division that results into 4 dissimilar or unidentical cells. So that's meiosis. Remember in grade 10, you learned about mitosis. What is mitosis? Mitosis is a cell division that results into 2 identical cells. So it's very different from meiosis. How is it different? Mitosis results into 2 identical cells, whereas meiosis results into 4 unidentical cells. Right? And another difference between mitosis and meiosis is that in mitosis you have 1 cell division, whereas in meiosis you have 2 cell divisions. Right? So those are the basic differences between mitosis and meiosis. Does that make sense? Okay, I hope it makes sense. So let's look at, we've already defined what is meiosis, right? We already defined. So here they're saying, the second column, they're saying that before meiosis begins, DNA replication takes place. This results in each chromosome consisting of 2 chromatids joined by a centromere. So let me explain the statement. So the first phase before meiosis even begins is interphase. So that's where DNA replication takes place, right? So DNA replication is that you are replicating, right, your DNA strand, right? And after you're done replicating, after a cell is done replicating the DNA strand, you result into 2 chromatids, right, joined by a centromere. A centromere just means that it's a point where 2 chromatids meet. It's a point, just the point where two chromatids meet. It's not a structure, it's just a point where two chromatids meet, right? So before meiosis, you have interphase. So what happens during interphase? You have your DNA replication, all right? I hope that makes sense. So let's look at the first division. So here they're saying that the first division, right, one diploid cell, diploid, remember, what does diploid mean? It means that you have two sets of chromosomes. Two sets of chromosomes. With regards to humans is 46. Does that make sense? So whenever they say diploid, just means two sets or 46 chromosomes. Okay, so one diploid cell gives rise to two haploid cells. Two haploid. So diploid, from diploid to haploid. Haploid is just one set, right? With regards to humans, right? In humans, it's 23. So haploid, when it comes to humans, it's 23. 23 chromosomes, right? So one diploid cell in the first division, one diploid cell gives rise to two haploid cells. So it means that we halve the number of chromosomes. So if you're starting with 4, you're ending with 2. If you're starting with 8, you're ending with 4. If you're starting with 10, you're ending with 5. So we're halving the number of chromosomes, right? Okay, let's go to the second division. So the second division states that the two haploid cells divides further to form four haploid cells or gametes, right? So the two haploid cells divide to form four haploid cells or gametes, right? So we see with the number of chromosomes does not change when it comes to the second division. So you start with two haploid and you end up with four haploid cells. So the number of chromosome does not change. But what changes? The number of cells. So the number of cells changes. So from 2 cells to 4 cells, right? But the chromosome number does not change. So let me just demonstrate this, make it an example. So if you're starting with 4, right, chromosomes, you end up with 4 chromosomes. So it does not change. So you end up with 4 chromosomes in each cell or in each gamete. A gamete is just a sex cell. I hope it makes sense. When it comes to males, we have a sperm. When it comes to females, we have an ovum. Does that make sense? Okay. Now let's go to the biological importance of meiosis. So it says here, crossing over introduces genetic variation. Genetic variation, right? These are distinct differences that exist within organisms of the same species. So these are distinct, right, differences that exist within organisms of the same species. So the reason why in your family you're not the same is because of genetic variation, right? It's because of meiosis. Meiosis introduces that genetic variation. I hope it makes sense. It ensures that sex cells have a haploid number of chromosomes. So it ensures that sex cells have a haploid number of chromosomes, thus maintaining the chromosome number, right? So just imagine if meiosis did not exist as a process, then it will lead to a catastrophic disaster, right? So it means that you have a sperm that has 46 chromosomes and an ovum that has 46 chromosomes. And those gametes will mix together and you have an enormous number of chromosomes, right? I hope that makes a lot of sense. Okay, now let's go through the terminology of meiosis. So homologous chromosomes. What are homologous chromosomes? So homologous chromosomes are chromosomes that are involved in meiosis, right? And they're the same in size, shape, and they code for the same characteristic. So they're the same in size, shape, and they code for the same characteristics. Hence homo. Homo means the same. So homologous chromosomes. So let's go to bivalence. So bivalence, what are bivalence? So bivalence are two chromosomes, right, that are involved in crossing over. So they are homologous chromosomes, right, to simply put it. But when they're involved in the process of crossing over, we call them what? Bivalence. Bi means two. But after crossing over, we no longer call them bivalence, we call them homologous chromosomes, right? Okay, chromosome. So what is a chromosome? A chromosome is a structure that contains, right? It's a condensed structure, right, that contains a DNA molecule inside of it, right? When I'm saying condensed structure, it's a condensed structure, right? It means it's a crystallized structure that contains a DNA molecule inside. Okay, let's go to our next term. Our next term is chromatids. So chromatids, right? They are just strands of chromosomes. There are two strands of chromosomes, right? Two strands of chromosomes that form one chromosome. Okay, let's go to centromere. What is a centromere? Centromere is a point at which two chromatids meet, or is a point at which two chromatids join, right? Okay, let's go to our next term here. Next term is crossing over. So what is crossing over? Crossing over is a process whereby uh, chromatids of different homologous chromosomes exchange genetic material, right? So it's where they overlap, and the point at which they overlap is called what? The chiasma or the chiasmata. After that, they exchange genetic information or genetic material. Does that make sense? So that's crossing over. So when two chromatids of different chromosomes or homologous chromosomes exchange genetic material. Okay, let's go to the chiasma or chiasmata. So chiasma is the point at which two chromatids of different homologous chromosomes overlap, right, during crossing over. It's where they overlap. I hope that makes sense. Let's go to karyotype. So what is karyotype? So a karyotype is is a representation, or some other people will call it a karyogram, right? So this is a diagram that shows all the chromosomes within a cell. So it's a representation of all the chromosomes within one cell. Okay, let's go to non-disjunction. What is non-disjunction? So non-disjunction, right, it's non-separation. It's when chromosomes, they fail to separate during anaphase 1 or anaphase 2. So it's when your chromosomes, they fail to separate during anaphase 1 and anaphase 2, or anaphase 2. Let me just say or anaphase 2, right? What is karyokinesis? So karyokinesis is the separation of the nucleus. So it's when the nucleus splits, right? The splitting of the nucleus. Let's go to cytokinesis. So cytokinesis is the splitting of the cytoplasm. Okay, let's go to the phase before meiosis starts. What happens in the phase before meiosis starts, right? So interphase. So this is what happens. DNA replication occurs. So you start with one DNA strand and you end up with two identical DNA strands. So we went over DNA replication in DNA, right? So as you can see here, you're starting with one DNA strand or one chromatid, right? This is a chromosome on it, like in its own, right? It's a chromosome, but we'll call it chromatid for purposes of this lesson, right? So you're starting with one chromatid and you're ending with two chromatids, right? And those two chromatids, they meet at a point called what? A centromere. They meet at a point called a centromere, right? The point at which they meet is called what? A centromere. Okay, now let's go through the points here. Each chromosome will now consist of two chromatids joined by a centromere. Okay, next point, DNA replication helps to double genetic material to be shared equally during each division, right? So it doubles genetic material so that it can be shared equally, right, in during cell division. So that each of the cells at the end of meiosis have equal, right? They have equal genetic material. I hope that makes sense, right? Because if DNA replication did not happen, then those gametes at the end of meiosis wouldn't have equal genetic material, right? Okay, let's go to the same point. It says unreplicated chromosomes, single chromosome as it appears in a cell before DNA replication takes place. So unreplicated, so this strand of this chromatid is called an unreplicated chromosome, right? Hence, I said it's called a chromosome. It's just called an unreplicated chromosome. Okay. Replicated chromosome, then here it says replicated chromosome, double chromosomes with two chromatids after DNA replication. So A double, this is just a double chromosome. This is an unreplicated chromosome. This is a replicated chromosome, right? But they're still chromosomes. I hope it makes a lot of sense. Yeah, I'm sure you might be asking yourself, what are these A, B, A, B? So these, we call these genes. They're called genes. Does that make sense? They're called genes. So A, B, C are called what? Genes. What is a gene? A gene is just a segment of a chromosome that codes for a particular characteristic. Now let's go to the phases of meiosis. Phases of meiosis. Let's start with, I always refer, or I always use this acronym, right? IPMAT or PMAT. You start with P because we've already passed right interface. You can say IPMAT or PMAT, right? So the first phase is prophase one, and remember, you use one or two when it comes to meiosis, right? You don't just say prophase. It's not mitosis. You don't just say prophase. Okay, let's start with prophase one. So let's look at prophase one. So prophase one, as you can see here, right? What happens during prophase one? In prophase one, we have the process of crossing over. So your homologous chromosomes or your bivalents, they'll start to cross over. They'll start to exchange genetic material, and your nuclear membrane or nuclear membrane, which is the membrane surrounding the nucleus, it starts to disappear. So it starts to disappear, right? Starts to disappear. And your centrosome, right? Your centrosome starts to split, and when it splits, it forms spindle fibers within it. As you can see, this is a centrosome. It splits, right? And when it splits, but it splits into two centrioles, and the centrioles will form spindle threads or spindle fibers within them. Does that make sense? I hope it makes sense. And also another point that you have to remember about prophase one is that the nucleolus, it disappears also, right? So the cell itself breaks down these parts. It breaks down your nucleolus, so it disappears, and your nuclear membrane disappears. It starts to disappear. Then let's go to the second phase. Second phase is metaphase one. So in metaphase one, our nuclear membrane has already disappeared. Our nucleolus has already disappeared. Right. The only thing we have is our centrioles at opposite poles and our spindle fibers. So our spindle fibers, they hold our homologous chromosomes. Right. So they'll hold our homologous chromosomes in the equator, right? So our homologous chromosomes, they line up in the equator randomly. We call that process random arrangement, right? Which is another process that brings about genetic variation. I hope that makes sense. Now let's go to anaphase one. Our next step is anaphase one. What happens during anaphase one? Right? Our chromosomes, homologous chromosomes, they are pulled to opposite poles. They're pulled by what? They're pulled by spindle fibers. As you can see here, the spindle fibers are pulling them away, right, from the center, are pulling them away from each other, right, into opposite poles. So homologous chromosomes are pulled, right, into opposite poles, as you can see here. Then we're done with our anaphase one. We go to our telophase. So telophase, that's where we have our cytokinesis, right? We have our cytokinesis. Our cytoplasm, it starts to split, right? It starts to split. I hope that makes sense. So we have in telophase one, we have our cytokinesis. Our cytoplasm, it starts to split, and it forms this part here, and we call this part a cleavage furrow, right? Call it what? A cleavage furrow. Okay. Now let's go to meiosis two. So meiosis two, we're starting with prophase two, right? Prophase two, we already have two cells, right? That have already split. So we have two cells in prophase two, and we do not have, right, crossing over in prophase two. We do not have crossing over in prophase two. Remember that we do not have crossing over, right? Whereas in prophase one, we have crossing over. But when it comes to prophase two, we do not have crossing over. I hope that makes sense, right? I hope that makes a lot of sense. Now let's go to metaphase two. So our metaphase 2, what happens in metaphase 2 in each of the cells, right? Remember, this process happens in two cells. So what happens? So our chromosomes, not homologous chromosomes, in metaphase 2, our chromosomes, they line up in the equator singly. Whereas in metaphase 1, homologous chromosomes, they line up in the equator in pairs. I hope that makes sense. So I'm already telling you the differences between metaphase 1 and metaphase 2. Okay, now let's go to anaphase 2, right? Anaphase 2, what happens in anaphase 2? Chromatids are pulled apart to opposite poles and they are pulled by the spindle fibers because as you can see, the spindle fibers are contracting during anaphase 2, right? Whereas in anaphase 1, chromosomes are pulled apart. In anaphase 2, chromatids are pulled apart, right? Okay. Then in telophase 2, you have what? Four cells, four dissimilar or unidentical cells. And also in telophase 2, you have cytokinesis in each of the cells. Does that make sense? Whereas in telophase 1, you ended up with two dissimilar cells whereas in telophase 2 now you are resulting into four dissimilar cells. I hope that makes sense. Okay, let's continue. The nicest thing is that you can rewind this video over and over again if you didn't hear me. Right, that's the nicest thing about this video. Okay, so we're done with the stages of meiosis. We're done. Okay, now let's go to, so there are questions here that are here. Okay, it's fine. Just go back here. Yo. Okay, just a moment. Ish. What am I doing now? What am I doing? Okay, let's go to non-disjunction, right? So remember, we said non-disjunction is the failure of chromosomes to separate. So chromosomes are failing to separate. So let's go to the first point. So the first point states that spindle fiber fails to separate during meiosis. So spindle fiber fails to separate during meiosis. Maybe they meant it fails to contract during meiosis, right? And the chromosomes, as a result of that, they fail to separate, right? And if they fail to separate during anaphase 1 or anaphase 2, that results into Down syndrome. We'll get to the point. We'll get to that point how it results to Down syndrome, right? Okay. So members of one pair of homologous chromosomes fail to become separated during anaphase 1 and may happen in anaphase 2. So this process can either happen in anaphase 1 or anaphase 2. Okay, let's go to the next point. If there is a non-disjunction of chromosome pair 21 in humans, it leads to the formation of an abnormal gamete with an extra copy of chromosome 21. Yes, it leads to an extra copy of chromosome rather, I might say, chromosome 21, right? Okay, and the next point says that two gametes receives extra copy of affected chromosome, two gametes lack that chromosome, right? And the state in which or where the state where two gametes receive an extra copy of an affected chromosome, we call that what? Trisomy, right? When each gamete receives an extra copy, we call it trisomy. And when each gamete receives or does not receive any chromosome, we call that monosomy. So we mean that that gamete did not receive any chromosome, right? Okay, now let's just try to demonstrate this and see this in a diagram, right? Okay, so this is a cell, right? So this is a diploid cell. This is a diploid cell, right? We start with two uh, chromosomes. Does that make sense? Two chromosomes. And you can see that there's a normal separation here. There was a normal separation in these cells, right? You have one here chromosome, and you have one chromosome. And in these cells, right, there was an abnormal separation here. One cell received one extra chromosome. What do we call that state? Trisomy. And one cell did not receive any chromosome. What do we call that state? We call it what? Monosome. I hope that makes a lot of sense, right? I hope that makes a lot of sense. Okay, now let's go to meiosis 2. So in meiosis 2, if non-disjunction happens, is that one cell, right, will receive an extra copy, right, of a chromosome, and one cell will receive no chromosome. Does that make sense? No chromosome. I hope it makes a lot of sense. Okay, here there was a normal separation in the cell, right? So this chromosome split into two chromatids. So there was one chromosome here, right? And there was one chromosome here. Obviously 23 when it comes to humans and 23 here, right? Hence they're saying 23, 23. Okay? And here there was a, obviously remember this cell, right, was also did not separate, right? It received an extra copy of a chromosome. So if it received an extra copy of a chromosome, it means that the cells that you have at the end of meiosis 2 will have extra copies of a chromosome. So you result with 24, 24. Here you just have 22, 22 because you did not receive any copy of a chromosome during meiosis 1. Does that make sense? Does that make sense? I know it's kind of confusing, but the more you practice it and you learn it, it will start to make sense, right? Start to make sense. Oh, let me start over again. Let me just try to explain this again, right? Okay, so you're starting with 46 chromosomes with regards to humans. Here it's just two. Let me just put it in the case of humans, right? You're starting with 46 chromosomes. Obviously, you know that when at the end of meiosis, eventually you have 23, right? We are expecting to have 23 chromosomes, right? Half of 46 is just 23. So 46 is just a diploid number, and 23 is a haploid number of chromosomes, right? So this is a normal separation here. So let's look at these two cells. Normal separation, and you see normal separation here, right? Where each cell receives 23 chromosomes and 23 chromosomes, right? It's fine. Now let's go to the non-disjunction part. So non-disjunction part, right? Can you see these two cells, right? So each cell received an extra copy. So it means that this cell will have 24, right? 24 chromosomes because it received an extra copy. And the other one, it means will have 22 chromosomes because it did not receive an extra copy. Does that make sense? What happens in meiosis 1 will affect what happens in meiosis what? 2, right? Does that make sense? Okay, now let's go to this cell. I hope you are following my pointer here. Let's go to this cell. Can you see this cell? Here. Can you see this cell? Right, look at the chromosome. So there was non-disjunction here. Obviously, it has 23. It has 23 chromosomes, right? But because now it receives an extra copy, it will have 40. It will have 24 rather chromosomes because it received an extra copy. Because this one did not receive an extra copy, it will receive 22. It will get 22 chromosomes, right? Okay. Here there was a normal. Here, let's go to this cell. Here there was just a normal separation of chromosomes, right, or chromatids, right? This cell, this gamete received 23 chromosomes, and this gamete received 23 chromosomes. Can you see that the chromosomes are equal in that case, right? Now let's go to this scenario here, right? Remember here in this cell there was non-disjunction, right? You ended up with 24 chromosomes. So it means if you have 24 chromosomes in meiosis at the end of meiosis 1, you also have 24 chromosomes at the end of what? Meiosis 2. So remember, from meiosis 1, right? In meiosis 2, the number of chromosomes does not change. So if you're starting with 24, you end with 24 chromosomes. So the number of chromosomes does not change unless the non-disjunction happens in anaphase 2, right? I hope that makes sense. So same as this one. This one had in meiosis 1, at the end of meiosis 1, it had 22 chromosomes, And at the end of meiosis 2, it will also have 22 chromosomes because the number of chromosomes does not change in meiosis 2. It only changes in meiosis 1. Okay, now let's go to reproduction in vertebrates. So this is just, guys, this is just a revision of term one, right? It's just a revision of term one, right? And the students that I teach already have notes to compare, right? They already have notes to compare with this presentation I'm making now. Okay, let's go to reproduction in vertebrates, right? So what to focus on? So these are things that you should focus on: external and internal fertilization, right? The two types of fertilization. And also, you need to focus on ovipary, vivipary, ovovivipary, right? So these are called reproductive what? Strategies, right? And also you need to know the structure of the amniotic egg, right? And also the extra embryonic membranes surrounding the embryo. Okay, so the next thing that you need to focus on is precocial and altricial development. Does that make sense? Precocial and altricial development. And also, the last thing that you need to focus on is parental care. Okay, let's continue. Let's continue. Let's continue. Wait, okay. Share. External fertilization. So, what is external fertilization? What is fertilization before we get to external fertilization? So, external fertilization is the fusion of the nucleus, right? Nucleus, remember, and also pay attention to this. I'm saying nucleus, not the sperm, right? Not the gametes. The gametes, they don't fuse together, but the nuclei fuse together, right? So, the nucleus of the sperm fuses with the nucleus of an egg or of an ovum. Right? So it's a process whereby there's a fusion between the two nuclei. Does that make sense? Between the two nuclei. So the nucleus of a sperm fuses with the nucleus of an ovum. So they fuse together. So that's fertilization, right? What is external fertilization? So external fertilization means that the fusion of the two nuclei happens outside the female's body. So it happens outside the female's body, right? Now let's look at the advantages and the disadvantages of external fertilization. So let's start here. So no additional energy is needed for the parental care or formation of the protective layer, right? So that's an advantage, right? No additional energy is needed for the parental care or formation of a protective layer, right? There's no energy that needs to go towards that. Okay, what's another advantage, right? There's no need for a male to have a special organ to insert the sperm into the female's body. So there's no need for a male to have a special organ to insert the sperm into the female's body. Okay, the final point says that chances of fertilization are enhanced by courtship display by fish. So chances of fertilization are enhanced by courtship behavior, right? So courtship behaviors are different, right, in different animals, right, or in different vertebrates, right? Okay, now let's go to the disadvantages. So the disadvantages are that chances are very slim for a sperm cell to meet the ova, right? Because it's happening outside the female's body. So that gamete can be intercepted by predators. So it can be intercepted by predators. Okay, let's go to another disadvantage. Another disadvantage states that predators eat the sperm and ova before fertilization can occur. Okay, so this is the point I've alluded to, right? Okay, let's go to another point. So another point says that strong currents carry sperm cells away from fertilization, right? So anything currents, right? There might be wind, there might be strong currents that could sway the sperm away from the ova or from the ovum. Okay, and the last point is that reproduction must take place in water, otherwise the gametes or fertilized ova will dehydrate. Right, that's why water is so important in the process of external fertilization. And also they have to produce a large amount of sperm for fertilization to be successful. Okay, so this is fertilization. So this is the process of what? Fertilization. So it's when the nucleus of a sperm fuses with the nucleus of an egg or of an ovum to form what? A zygote, right? So a zygote is just a diploid cell. I hope that makes a lot of sense. Let's go to internal fertilization. Let's go to internal fertilization. So internal fertilization, what is internal fertilization? So it means that the fusion of the two nuclei happens within the mother's body. When I say two nuclei, I mean the nucleus from the ovum and the nucleus from the from the sperm. So it means it happens within the mother's body. Right now, let's look at the advantages and disadvantages of internal fertilization. So this is the first point. Ensures that the sperm cell come into contact with the ovum. Right. So that's the advantage. So it ensures that the sperm cell comes into contact with the ovum. So it's a showcase because it's happening within the mother's body. Okay. The second point is that developing embryo is protected from predators and removed from harsh conditions. Obviously, it's happening inside the mother's body, so it means that the developing embryo and the gametes and the gametes are protected from the predators, so they cannot be intercepted by predators unless the predators they eat the mother, right? So there are higher chances of fertilization as compared to external fertilization. Okay, now let's go to disadvantages. Fewer eggs are produced. The reason why fewer eggs are produced is because it's happening inside. So there's no threat from predators. Does that make sense? Okay, another point is that the animal must have copulatory, right? Copulation. Copulatory comes from the word copulation. What is copulation? Copulation is sexual intercourse, right? Copulatory organ to insert sperm cells into the female's body. So the animal, that's a requirement, right? Because the sperm cell needs to be inserted inside the female's body. I hope that makes a lot of sense. Okay, let's go to embryonic development. Embryonic development or reproductive strategies, right, that are used by vertebrates. Right, let's start with ovipary and ovovivipary, right? So what is ovipary? So ovipary is that eggs are laid outside the mother's body. So eggs are laid. So ov means egg. So it means that eggs are laid outside the mother's body. Then ovovivipary, the eggs are retained inside the mother's body until they hatch, right? And the young is born live, right? So they hatch inside the mother's body, whereas ovipary, they hatch outside the mother's body. I hope you can see the difference between ovipary and ovovivipary. Ovipary means that the eggs hatch outside the mother's body, and ovovivipary, they hatch inside the mother's body, right? The eggs are retained inside the mother's body, and the young is born alive. Okay. So now let's go to the advantages. The advantages, right, is that eggs and sperm cells are produced in large numbers to increase chances of survival to adulthood, right? So that's the first one, first advantage. Parental care ensures survival to adulthood. Okay, let's go to disadvantages. The young one is protected from cold and predators to ensure survival. The young are well developed when born, therefore their chances of survival increases because they will be able to evade their predators. Okay. Okay, so those are advantages and disadvantages of ovovivipary and vivipary. I've already explained what is ovovivipary and vivipary and the differences between the two. So these are advantages and disadvantages of ovipary and ovovivipary, right? I hope that makes a lot of sense. Okay, let's go to vivipary. So what is vivipary? Vivipary is that the young, right, is protected in the mother's uterus, right? The embryo develops in the mother's uterus and receives nutrients from the placenta. So the young is in the mother's body. So we also, as humans, we display viviparous, right, reproductive strategy. Okay. As you can see here, the picture here. So this is an elephant, is a young elephant, right? It's within its mother's body and it's receiving nutrients from the mother's placenta. Okay, let's start with the advantages. So the advantage is that the development occurs within the mother's body, ensuring protection of the developing young. So inside the mother's body, right? Let's go to the second point. The second point states that the young are born well developed and are therefore less prone to being attacked. So they're born well developed, right? As I'm sure you know that young elephants can walk, right? They display precocial development. They are born with eyes open, right? They can walk, they can evade predators and everything. So that's what this point means. Let's go to the last point. The last point says that parental care increases the chances of survival of the young, right? So it increases the chances of survival of the young, right? So the parental care is very high, right? Before and after birth. Okay, let's look at the disadvantages. The disadvantages are that number of offspring produced is few, right? Because only a few, right, a few amount of offsprings can fit into the uterus. Hence, the number of offsprings are few, right? Because the uterus is not that big. Okay, more energy is used to provide parental care, so they use more energy. The mothers use more energy to provide for the young. Okay, let's continue. Let's go to the amniotic egg. So, the amniotic egg. So as you can see that the amniotic egg is surrounded by 5 layers. I always use the acronym ACAs, right? So the A stands for amnion, right? The C stands for chorion, right? And the A stands for allantois, the Y stands for yolk sac, and the S at the end stands for shell. Okay, now let's read. So the first point says that the embryo develops inside the amnion and is protected by many membranes and a hard shell, right? So those membranes that are protecting the embryo, as you can see here in this picture, there's an embryo, and the embryo is protected by 5 membranes, right? And those membranes are called, I'm also including the shell as a membrane because it adds to the protection of the embryo, right? So, and those membranes are called extra embryonic membranes because they're outside the embryo, right? They're outside the embryo. Does that make sense? Okay, let's go to the second point. Amniotic fluid protects the embryo from drying out. Obviously, within the embryo is swimming there or it's free flowing within a fluid called the amniotic fluid. And that amniotic fluid prevents the embryo from drying out. Some other books will call that desiccation, right? It protects the embryo. It allows the embryo to move freely within that fluid, right? And it also prevents the embryo from mechanical shock, right? So it's a shock. That amniotic fluid becomes what? A shock absorber, right? Okay, so let's go to the next point. The next point says that it feeds from the yolk, right? Okay, so before we explain this point, let's start explaining the membranes that are surrounding, right, the embryo, right? So the first membrane surrounding the embryo, you have the amnion. As you can see, the amnion. So what's the function of the amnion? The amnion encloses the amniotic fluid, and also it secretes. It's responsible for secreting the amniotic fluid. So that's the function of the amnion. It's the first membrane, right? And the second membrane here is the chorion, as you can see here. What's the function of the chorion? The function is responsible for gaseous exchange. So it allows oxygen to go into the embryo and carbon dioxide to come out of the embryo, right? So it's responsible for gaseous exchange. Oxygen in, carbon dioxide out, right? And the next membrane is you have your allantois. So this bag-like structure, right? This bag-like structure is called what? Allantois. What's the function of allantois? Is that it stores waste. So it stores waste, right? And the last one, or the second last one, is your yolk sac. And you see the difference in size and shape because I know that a lot of people might be confused when it comes to allantois and yolk sac. This is the yolk sac or the yolk, right? So what's the function of the yolk? So the function of the yolk is that it provides nutrition, right? Nutrition for the embryo, right? And then lastly, you have your shell. Obviously, your shell serves as a protective shield, right? It serves as a protective shield for the egg. Okay, let's continue. I've already explained most of the points that are here, so we won't go through them, right? Let's go to precocial and altricial development. Precocial and altricial development. But before we continue, I think that we should end here and wait for part two, right? Part two. From part two, I'll continue from here to human reproduction, right? I'll send part two just maybe tomorrow. Tomorrow, yes, tomorrow or today. I mean, I might just upload part two today, right? Okay. Good luck, guys, on everything that you will be writing in life sciences, whether it's a test or whatever you be writing that regards life sciences. Good luck. Good luck, guys. All the best.